Howdy, and welcome to Respiratory Zone 1, the respiratory membrane. So, in the previous podcasts, we did the conducting zone part 1 and the conducting zone part 2. With the respiratory zone, we just have a part 1. So, if you look at the bottom of the slide, you can see on the left side there's a lung, and that blue structure going into the lung is the left primary bronchus, and then you can see the blue structure branches into a secondary bronchus, and then those secondary bronchi branch into tertiary bronchi, and the bronchi then, as they get smaller and smaller, they become bronchioles. And then if you look at the right-hand side of the cartoon, you can see there's a terminal bronchiole and respiratory bronchioles. Though the terminal bronchiole, as you can see, doesn't have these outpouchings of the alveoli, and that's where you do gas exchange, is at the level of the alveoli. So the terminal bronchiole is the end of the conducting zone, and the respiratory bronchioles, that marks the beginning of the respiratory zone, because that's where you can do gas exchange with oxygen and carbon dioxide. So this first bullet tells you that the respiratory bronchioles, you get some outpocketing of alveoli for gas exchange. Within the lumen of the respiratory bronchioles, the epithelium is simple squamous, and there's no more cilia. So you don't, at this level of the respiratory bronchiole, you don't want any mucus or cilia or anything that can interfere with gas exchange. So that's why you don't have cilia and mucus at the level of the respiratory bronchiole. From the respiratory bronchiole, you go into an alveolar duct. So if you look at the cartoon, the way you can tell the difference between a respiratory bronchiole and an alveolar duct is the alveolar duct has a higher density of alveoli versus the respiratory bronchiole. But like I said, wherever you find alveoli, you can get gas diffusion, oxygen and carbon dioxide. And in this cartoon, it doesn't show, but there's a mesh of capillaries covering each of the single alveoli in the cartoon. So there's about 300 million alveoli in your lung. So that means there's a huge SA, or surface area, for gas exchange, which is what you want in a mammal that breathes air. You want a huge surface area to allow for oxygen to diffuse from the alveoli into the alveolar capillaries, and for carbon dioxide to diffuse in the opposite direction. In a person who has emphysema, they've lost the alveolar surface area, and they've also lost the elastic tissue that lightly surrounds the base of an alveoli. So when you lose your alveoli, you lose the ability to do gas exchange, and when you lose elastin, you lose the ability to recoil your lung during exhalation. So people with emphysema, they have to work hard to breathe the air out, or they have to work hard to exhale. So they often have a barrel chest. The alveoli themselves are connected by channels and pores, so that if one alveoli or one alveolus becomes destroyed, airflow can go from that non-functional alveoli to another, or that non-functional alveolus to another alveolus. So this is a cartoon that shows a alveolar sac. So on the left-hand side of the cartoon, you can see there are several alveoli. In the center of the cartoon on the left side it says alveolus, so that's just the space in the alveolus. And then you see those pink sort of polyhedral shapes in the alveolus. Those are actually type 1 alveolar cells, and that's where gas exchange occurs. That's where oxygen and CO2 can diffuse across these type 1 alveolar cells to get into the alveolar capillaries. So continuing on the left side of the cartoon, there's a type 2 cell that secretes surfactant, and surfactant is a molecule that breaks up water tension so that your lungs don't collapse. So if you had purely water environment in your lungs, the hydrogen bonding between the water molecules would cause your lungs to collapse. But because you have type 2 cells in your lungs secreting surfactant, the surfactant breaks up those hydrogen bonds between water molecules so that your lungs don't collapse. So also in the cartoon, if you look at the left side and up on the right side of the left-hand side of the cartoon, it says there's an epithelial cell nucleus. So the histological type of cell in the alveolus is an simple squamous epithelial cell, but because that simple squamous epithelial is in the lung, it's called a type 1 cell. There's also macrophages within the lungs in the alveoli, and if you recall from the immune system, the macrophages engulf foreign invaders and destroy them. So because this is a cross-section of an alveolar sac, if you look at the left-hand side of the cartoon where there's a black circle, they're showing you the fused membranes of a type 1 alveolar cell and an endothelial cell in a capillary. And then if you follow that gray arrow to the right-hand side of the cartoon where they enlarge that fused basement membrane, you can see in that circle you have the alveolus, or the space, and then you have a type 1 alveolar cell, and then you have a fused basement membrane, that yellow stuff. It says it fused basal lamina. For our purposes, a basal lamina is the same thing as a basement membrane, and that basement membrane is shared between the type 1 alveolar cell and an endothelial cell of an alveolar capillary. So that is the site of gas diffusion. So that's called the respiratory membrane. And you can see in the cartoon on the right, oxygen is diffusing from the alveolus into the alveolar capillary, and CO2 is diffusing from the alveolar capillary into the alveolus to be subsequently exhaled. I told you that the type 2 cell secretes surfactant to break up the water tension. It also can secrete antimicrobial proteins for any microbes that get that far into the respiratory tree. Just a little fun fact for dinner conversation. You clear, so you either expectorate or you swallow about 2 million dead macrophages per hour. So your macrophages are really working overtime out in the periphery of your lung in these alveoli to destroy foreign invaders. So, and then on the back to the right-hand side of the cartoon, it says that the alveolar capillary, upstream of the alveolar capillary, is the pulmonary artery. So remember, that's bringing, pulmonary artery is bringing oxygen poor, CO2-rich blood to the lungs. And then I have a question: how many cell membranes does oxygen cross before it finds hemoglobin? How many barriers? So answer that question, and then we'll talk about that in class. So this slide doesn't have any cartoons on it. It's just basically you can read through it, but I've talked about a lot of these concepts. The type 1 cell histologically is a simple squamous, but because it's in the lung, it's called a type 1 cell. It's part of the blood-air barrier. The gases, primarily the gases we're interested in, are oxygen and CO2. They diffuse down a pressure gradient. Oxygen will move down its pressure gradient from an area of high pressure oxygen to low pressure oxygen, regardless of what CO2 is doing. That's called oxygen's specific partial pressure. That respiratory membrane is very thin. So just the type 1 cell, if you think of a sheet of paper, it's 15 times thicker than a type 1 cell. Type 2 cells we talked about in the previous slide. They secrete surfactant to break up the surface tension. And we talked about how water molecules attract each other via hydrogen bonds within the alveolus, and the surfactant breaks up those hydrogen bonds so that water doesn't collapse the alveoli. And premature babies, they don't have surfactant, so they have to be put on a ventilator. And then the alveolar macrophages, because they're macrophages in the alveoli, they get special names called a dust cell. And we know that white blood cells and macrophages specifically phagocytize and digest foreign invaders. And this is the last slide. I really like this slide. This is a scanning electron micrograph of a capillary bed covering alveoli. So the gray balls are alveoli, and then the red, you can see an arteriole is bringing in this network of capillaries, and you can see how the capillaries enmesh the alveoli, which is what you want for ideal gas exchange. And those gases we're interested in, of course, are oxygen and CO2. And that's the end.